Yeah. Welcome to the meeting. We have Himanshu here, and Morgan, Dick, and Susan. I guess Himanshu, can you give an update on your things? I don't know if Sushmanth will be here today, but
Yeah, hello, I'm audible.
yeah.
Okay, I'll start. Is my screen visible?
Yes.
Yeah. Yeah, okay, so I'm still continuing my work from last week about the nucleus segmenter and the membrane segmenter. So basically, like my main issue with collab was the memory had but the free resources were not there. Like finished all my compute, so I changed my workflow from Colab to Kaggle to just train the model, and like I got I tried to change some things and finally got it running on Kaggle as well. So basically, right now I changed a few things in the code. The I found another way of getting the bounding boxes. There are two ways in which we can use from the masks, and we find the x min, x max, y min, y max position of all the coordinates of the box with respect to masks. And another way is to use PyTorch's own custom implementation, which also gives you the similar results. But this I found that it's better to use their function because it's more quicker and more intuitive. So and also one thing I tried to do was to train for a longer period of time. So the advantage of using Kaggle over uh, Collab is that Kaggle gives you around 15 to 16 GB of GPU for around 30 to 40 hours of usage per week, and it's much better than what Collab gives you, because the resources and the free resources get switched every month, get renewed every month in Collab, and here it's only 1 week. So I tried to train the model once again, and basically for more epochs, and I ran into a problem where my my learning rate changed to zero. Like it was zero and it was not learning further. So I tried to Google the error about it and to see how to change a few things so that it gets up and going. So one thing I noticed from this was it was only finding only one mask out of all the masks that are given. I'm not sure if I have an image. So basically, if these are the bounding boxes, it was only focusing on one bounding box. So the problem is with respect to gradients. So it's the gradients are vanishing from uh, for each epoch, next subsequent update updating the weights during training, the gradients are vanishing. So which is why you're only it's only focusing on one bounding box. And for so the trick to overcome that is to hyperparameter tune it much better than what I've been doing right now. So and also changing the optimizer and all. So I've used in this stochastic gradient descent, which is known to do that. So I also tried it with Adam, and in Adam, the gradients were exploding because the loss, for example, it's like 1.3, 1.2 something, and it just skyrockets. Like it goes till really big loss, like 5,000 something. So right now, the main thing I'm focusing on is in parameter tuning, and also optimizing the thing so that I overcome this. And also uh, I'm reading papers of the DevoGraph part of it. So there is a paper for using persistent homology in graph neural nets. Like, so it's doing link prediction, which is not like we are trying to do tracking, but the process before doing the final task, that is to generate the graph embeddings, it's kind of similar, and it's similar to what uh, it's similar to what we were discussing last week. So
Yeah.
like now I'm just looking into the what do you say? You could say the uh, papers part of it, like reading materials and all.
Right.
So after once that is done, and once my laptop actually starts working again, because like it has an issue right now. So I've come from my brother's laptop. So basically my CPU fans they stopped working like completely.
Oh, wow.
So I have to change them. Yeah.
Well, that's great. Let's go back to the paper. What is that paper from? Is it from a journal or it's archive? This is
Archive. Yeah,
yeah.
it's from.
Okay. Yeah, I'm just getting the number.
I'll send it in chat. Just a second.
Yeah. Okay, that's great. Yeah, we were talking about that last week. That'll be a good thing to go over. Yeah, and yeah, so that they're doing link production here, but the code you're working on now is for sort of segmentation, getting to that point of creating sort of links or creating nodes, and then maybe creating edges from that. So you're having problems with the gradient vanishing and the gradient exploding. So it's clear something is wrong there with the hyperparameters. So what's your strategy for solving that?
Basically, I've come up with like two approaches. One is to like kind of focus more on the data set, because like right now I'm taking all the images which was given, and since it was like a TIFF file which was changed into like some like more images, like we converted the TIFF file into PNGs, and it was like a series of images. So right now I'm trying to like take the best images which has the like kind of correct bounding boxes, which was and then to focus only on those images and try to fine tune it on that, and also remove like all the noisiness in the data, like some more and after that I'll focus more on changing these parameters and to see which one is the most perfect one. Like now I'm using the step LR. So for the LR scheduler, which basically changes the learning rate with respect to the epochs, and so I'll just play around with these and try to get better results. My main concern was the GPU problem, which was like in collapse. So right now I can do more iterations of the parameter tuning and everything, so it's better now.
Okay. Yeah. Well, that's good. Yeah, keep working on that, and you know, keep us posted. Hope your computer gets back to normal soon.
Yeah.
All right. Yeah. Thanks for the update.
Stop sharing.
Yeah.
Hi, Bradley.
Hi. How are you?
Sorry for fine.
That's fine.
Sorry for late. My laptop was also not working. I'm actually at the Apple store right now.
Oh, okay.
I came to repair it up. Actually, I talked with Mayuk. I have submitted my first evaluations today afternoon. He told that he will do my evaluation tomorrow or day after tomorrow. We will decide a time and I will let you know. If you are free, you can also add in the meeting and just see my progress. Right now, my progress was I almost implemented SAM because I was following one tutorial to implement SAM actually. This tutorial I am using SAM, fine tuning SAM using transformers library. I am using this guy's code on our data set to implement it. I'm almost done with it. Just a minute. I'm sharing my screen.
Oh, I'm not seeing it. Oh, there we go.
Okay. this is the transformers tutorial actually, which was to fine tune the SAM model. So I am following this tutorial to fine tune SAM because I have searched so many ways it was not implementing. So I was following this tutorial. I'm almost there to implement it because if you see here the results of ground truth mask, pixel values, etc. And this is my notebook which I have implemented. You can see that there are exact same values, these three values. I was getting same as they implemented. I just need to write a training loop, then model will be trained. I'm also having issues with GPU, so I bought some GPUs. Yesterday I bought some GPUs from Paperspace. Around it took $40 or something, so I took it. It was the cheap one. Then my told to start implementing it, and on the DevoNet also. Sorry. Okay, sorry, I have issues with my network. So tomorrow, if you are free, Bradley, can I have my evaluation tomorrow?
Yeah, I can. I guess they open today, or
I mean, if it's possible tomorrow, because I was at outside, I couldn't really have all these documents with me right now.
okay.
Maybe tomorrow I will show my whole update with Mike also gonna join tomorrow's meeting.
What time is it?
I will ask him and I will. According to your, can you give me some time slot? I'll discuss with Mike and I will just tell him to attend the meeting at that time.
Okay. Yeah, it's usually around this time is good.
Okay. I will let you know tomorrow.
Okay.
Almost he was okay with it. He saw my updates and all good. He told to just start fine tune SAM and show it to him. I'm almost there. My project is also over. And another thing is that there are so many things I want to discuss. Tomorrow's meeting I will discuss with you and Mayak.
Okay.
And hopefully Mayak will also join to that meeting.
Oh, that'd be good. Yeah, be good to see.
Regarding the paper, research paper and all these things, I just want to talk combinedly with all these because the DevoLearn was started at 2020 time.
Yeah.
From there onwards, there was no particular paper. Actually, there are journals, but there is no particular paper. So we are thinking to make a research paper out of it. So yeah, we'll discuss tomorrow in the meeting combinedly.
Okay. Yeah, that's good.
This week, all my updates I will update in a blog and I will just show it to you in a blog. I will keep that in the Slack channel.
Okay.
Sorry for this. Actually, this laptop stopped working, so I came to the Mac store to repair it.
Oh yeah.
Well, maybe tomorrow I will show the whole update.
Yeah, that would be good. Sounds good. All right, thanks for updating today.
I will leave the meeting now. Tomorrow I will show you the whole update.
Okay. All right, thanks. See ya.
Thanks.
So yeah, that was he was doing his update. So he's also using Kaggle notebooks, and we're working on that. And I'll meet with him and Mayuk tomorrow. I know, like, he's been working with Mayuk in a separate context, but we'll kind of bring those together tomorrow. So we'll see. Yeah. So any updates from Susan or Morgan or Jesse or Dick?
Yeah, I could. Well, I've had some discussion over mapping the differentiation tree to the genome.
Oh yeah.
Okay. Now we have differentiation trees for C. elegans, a partial one for axolotl. They're not poppy yet.
Yeah.
Okay. So C. elegans might be the place to look. Okay, so I'm going by a very simple premise that one, differentiation is represented in the DNA.
Right.
Okay. Two, that differentiation occurs by copying a group of genes and then having them diverge.
Yeah.
Okay, and that's what I called continuing differentiation. The ability to copy something responsible for differentiation over evolution, and duplicate it, and then the duplicates diverge from one another, producing two cell types where you had one.
Yeah.
Okay, that's the basic idea. Now, it's an untested idea. So, as I said in a note to you, the problem with finding the DNA responsible for differentiation might be approachable by saying, let's suppose that there's a motif in the DNA,
right,
which is similar for all steps of differentiation. Okay? If that motif, if we can find motifs whose count equals the number of nodes in the differentiation tree, then we have a possible candidate for the DNA that is responsible for differentiation.
Yeah.
So I'm not sure how to go any further than that, because obviously for DNA we don't know the number of base pairs involved in differentiation, and once we knew it, we don't know what varies in it and what's constant in it.
Right.
Okay, so this is kind of looking for a pattern that might match the differentiation tree. Now, the only clue that I can think of right now is that whatever the motif is, the number of copies of it should equal the number of nodes in the differentiation tree.
Right.
So you could imagine a massive long-term computation which creates motifs and then looks for them and counts how often they occur. And once it finds at least one that matches the number of nodes of the differentiation tree, then, well, we're not home, but we've got a candidate.
Right.
Okay, so I'm not sure how to proceed from there.
Yeah. Yeah, it's an interesting idea. I think we've talked about this in the past, but so
Yeah,
yeah.
It makes blasting look simple.
Yeah.
Okay, because in a sense it's kind of blasting, but I don't know. You got to assume there's going to be variation in the motifs.
Right.
Okay, and you're looking for a count of the motif in the whole genome.
Right.
Okay, so it may be computationally impossible right now. I don't know, but it's conceptually possible.
Right. So let me.
So I don't know how many of the people joining us today are familiar with blasting and stuff like that.
Yeah, let me draw it out a little bit. So, a differentiation tree is where we have, let me edit here. Okay. Differentiation tree is where we have a tree that diverges. So we have like a single cell type, and then it splits to 2 cell types, and then to 4 cell types, and we go down. Right. But we'll just use this as a quick example.
Okay.
Now, of course, DNA has a structure of base pairs, so you have these combinations, these codes that kind of come out if you were to take like 100 base pairs, or it'd be a code there of a sequence of different letters. Let's take like a 6 base pair code here. Let's say I'm just kind of making one up because just for the sake of simplicity. So this is a 6 base pair code here. Now, what you're saying is that this might be like involved in differentiation. It'd probably be longer than this because this probably doesn't give you much. Sometimes you get repeats of things. You get, tandem repeats, which are like 2 letters repeated. You get other types of repeats that are more complex, but they usually have some sort of functional significance. I mean, sometimes they don't, but so
you need a similarity measure of that to your guess.
Well, yeah. So you can have like you can basically go through and look for different motifs. If you know that if you see that, like you could design an algorithm to go through a draft genome and look for different repeats or different motifs of different types. If you predict that, because we know what the codons should look like, we know what the amino acids should look like that come from the codon. So we could say, well, we're interested in this combination of amino acids. And so that would give you a motif. And then you could see how many times you find that in a draft genome. So you could search for, say, these 6 bases at the top, and it would give you a number. So,
okay,
yeah.
One way to approach this might be to say, if for each cell type, you, how should I put it? For each cell type, there are unique genes produced if it's which distinguish it from other cell types.
Yeah. So a set of unique genes. Yeah.
Now, that's for the differentiation itself. The question is, what would they have in common?
Right.
If you pick a cell at the top of this graph and a cell at the bottom, what do they have in common with each other?
Yeah.
Now, the general metabolism is obviously one, but that doesn't help much. Okay, I'm trying to think ways through it. Yeah, the housekeeping genes.
Yeah, basic differentiation.
Okay.
Yeah.
Yeah, so we're looking for what is involved in differentiation, and can we make a motif for that, and then hunt for it, and then count the number of times it occurs.
Yeah.
If the number of times it occurs equals the number of nodes on that graph, then we have a candidate.
Yeah.
Okay, so
yeah, and also thinking in terms of go ahead.
Yeah, okay, it might be possible to do this, but finding what to look for, it's sort of like Carl, the stuff with Carl Woos, which ended up finding Alicea.
Yeah.
Okay, he had to find something that was good enough to be in common and evolve slowly and look for it. And he ended up with the S16 RNA from bacteria.
Right.
Okay. So he intelligently honed down on something that might have it.
Right.
There might be a way to do that for differentiation.
Yeah. So one thing, this kind of what I'm getting from this is that you have these sets of genes for each node, and I'm kind of drawing a Venn diagram over a tree. But so each circle is a set of genes that are involved in sort of differentiation. But then you should and there should be some uniqueness, but there should also be some overlap, say with the daughter cells.
Okay. Right. Maybe that's the way to look at it. Take any triple, a cell type, and then 2 cell types that are derived from it.
Yeah.
Okay. And then ask, what do they have in common?
Right.
That might hone in on the differentiation genes.
Yeah. So then, they should obviously have housekeeping genes in common, but we know what those are. We can throw those out. Then we have things that maybe are involved in differentiation. There are a lot of functional classes, but you could narrow it down probably.
Yeah, maybe. Okay. We might need some supercomputers. Hopefully we don't need a quantum computer yet.
Oh, no. The way this works generally is that you usually have a set of candidates. So people will do searches. They'll search for some string, or they'll search for a set of genes, or maybe different regions of the genome that they want to search for, and then they say, well, there's a certain probability that this is, a match in this area or this area. We know kind of from functional studies what different parts of the genome do, if it falls within a gene or not. And then also we know, we can also predict kind of its likelihood that it's,
Here we have to go a step backwards and say, well, what could it be?
Yeah, I mean, that's function is always really hard unless you have studies that tie to. I mean, they're probably, if we go back to some of the literature too, you can probably identify things that are involved in, differentiation. They have like a sequence, and then find motifs in those sequences or something like that.
Well, I think C. elegans might be the ideal organism because they're around 800 to 1,000 cell types.
Yeah.
We know how many nodes we're looking for.
Right.
Okay. And I don't know. Once this is done, some of the mutations might involve splitting some of these differentiation regions, and therefore screwing it up.
Yeah.
There are lots of mutations for C. elegans.
Right.
Some may involve proper new differentiation. Okay.
Yeah.
So it's a tough problem, but I think it's solvable.
Right.
Okay.
Yeah. So yeah, I mean, then the other problem too is like if you have different cell types, you're talking about the things that make them unique or the things that are involved in differentiation. Because I mean, a lot of people will go in and look at like different tissue types and know that there are differences in tissue types, and that's just a bunch of cells in the tissue that they sequence, and but those aren't involved in differentiation per se. Those are a different thing. So that's the other thing, is that you want to be specific to that. And it might not be that hard because a lot of things are just kind of like, this is specific to bone, this is specific to muscle. And it's really about, getting like upregulation of those genes more than like the actual.
An advantage of C. elegans is that every time there's an asymmetric division, you get two new cell types. Okay?
Yeah.
And you only have about 50 cases where it doesn't happen, where you get a pair of cells that are supposed to be similar.
Yeah.
Right. So that's why I think C. elegans might be the right organism. Hold on a second.
Okay.
Hello?
Well, that was good. So that's, I don't know if people are interested in that. If people out there want to work on that problem, we could talk. We've been trying to do something like this for a while, but we've been kind of, it's hard to kind of get it going and doing something interesting with it. So it's hard work because it's a lot of bioinformatics. Not everyone has that skill set, and
it's okay. It's just Dick pushing computers again to do more.
Oh yeah.
Yes. Yeah, I mean, conceptually, it's not that hard, but the combinatorics are immense.
Right.
Yeah. Okay, but given that we have the fully sequenced genomes, the data is available.
Yeah.
Okay. So that's why I think this would be a major step towards testing whether differentiation trees are the basis of differentiation, and if they are, okay, what's the representation of DNA?
Yeah.
Okay, because we have a strange situation of mapping a tree into a linear structure.
Right.
Okay, and that's why it's might be more complicated than I imagined. But okay.
Yeah.
But if you try to take a tree and map it into a linear structure, you'll see the problems.
Right.
Okay, branching structures don't become straight lines easily.
Right.
And yet, if DNA is the cause of differentiation, it's got to be there.
Yeah. Well, it's either going to be the same sequence, like sort of.
Oh, there we go. There we go again.
So yeah, it's usually like it's either going to be the same sequence that's conserved across different cell types, or probably more likely you're going to have maybe some changes depending. I guess if the differentiation process is the same, it would be basically conserved. So it would basically be maybe like an instruction of change to this cell type, or a lot of times you have genes that are for like things like stemness, or things that enforce the identity of a cell. So like a lot of times you'll have genes, well, there's a whole set of what they've called stemness genes, which maintain like pluripotency. And that's something that gets relaxed when you have different cell types. But there are also genes that are like definitive of a certain cell type. And so I don't, yeah.
You get stem cells in C. elegans.
Yeah, they do.
You do?
Yeah.
Okay. What happens when they differentiate? Do they differentiate further up the differentiation tree or to anything?
Well, in C. elegans, you have a deterministic differentiation pattern. So, certain cell types, they start from the one cell stage, and they're sort of developmental cells, which are basically pluripotent cells. Well, they're not pluripotent in the conventional sense, but they have this, they're not yet their adult terminal differentiated form until they get to a certain point, and then they differentiate. And it's like
Okay, I guess what I'm asking is, when they do differentiate, do they go up the tree or down the tree?
I guess down the tree.
If they go down the tree, then they can become something more basic, like all the way back to an egg, maybe.
Huh.
Okay, if they go up the tree, they can only form future types, which are getting closer and closer to the adult types of cells.
Oh, okay. Well, I guess probably down the tree then. I guess I'm not sure what the terminology is. Like.
Okay. The terminology is trans-differentiation.
What was it?
Trans-differentiation.
Oh, okay. All right.
Okay. So, it could be the whole idea is wrong. Maybe the thing to do is look at differentiation patterns of stem cells in C. elegans. See if they can be classified as to what part of the differentiation tree they produce.
Yeah.
If they can produce the whole tree, then maybe they can go all the way back down. If they can only produce future parts of the tree, then we have something quite specific.
Yeah.
I don't know, is there data available for that?
I don't think so. I mean, there's a genome, and there's like I think it's because I know they do a lot of single cell sequencing, so they may have single cell data available. So it'd be interesting to see what the differences are in the actual sequence. Now, what's probably what's easier to find is stuff like RNA-seq data, where you have like sequence, but you have things that are upregulated in a single cell, or downregulated in a single cell. So you actually have the sequence associated with that activity.
Okay.
And then that would actually be informative, because you could divide up the genome further into what's actively being transcribed or not.
Right. Okay. Yeah, in a way, this might involve the epigenetics of it, because if differentiation is caused by the DNA, then the cell types of the given organism could be represented by closing down more and more of the DNA until you get to a specific set.
Yeah.
Right. In which case, one would like to know the epigenome for a cell and subsequent cells from that, and compare them. That might be a way of getting at it.
Yeah. Yeah, that would be. Yeah, so there are a number of ways to get at this problem, and really it's going to depend on what kind of data we have available,
Yes.
and then what.
Yeah. Has anyone worked out the epigenome for C. elegans at all?
I don't. I think people have worked on like different things like methyl. I think methyl markers and things like that in the genome, but I don't know. People have also done things with epigenetic inheritance, and then associated with that work, they've done some work on looking at the epigenome, but I don't know where those data are. I'm sure those data are out in.
Okay, and do they map in any sense to the differentiation tree for C. elegans?
I'm not sure. I'd have to look at the data.
Okay. Well, that may be another approach to it.
Let's see.
Yeah, I think if we could solve this problem, it would be a major breakthrough.
Yeah, it would be great. I mean, I don't think people have. I think people think about it more in terms of like the cell type that's what's upregulated in the cell type, but that's kind of like beside the point. Is there a mechanism that sort of guides the cell towards a fate? So like obviously you have the cell and its associated things associated with that cell type. You have the stem cell and things associated with the stem cell, but what's that transition look like? And that might be where those.
Then you have the asymmetric division, and what is it that becomes different between the two symmetric divisions?
Yeah. I'm just writing some notes here.
Okay.
Right. But yeah, that'd be good. Well, maybe I'll take a look and see what kind of data there is for these types of things. I'm not really sure what the state of the art is.
It seems to me that the thinking about what causes differentiation is very muddled.
Yeah.
It's mostly gene circuits and whatnot. And what does that mean?
Right.
How about the cell state splitter and where it belongs in amongst all this?
Well, that's a possibility. If the cell state splitter is universal at all stages of differentiation, then the genes involved in the cell state splitter might be part of this differentiation apparatus.
I have a student group who might like you to come on and explain the cell state splitter.
Oh boy. Okay.
I tried the other day and I said, well, skip the first 7 pages of this, I don't get the genetics.
And when I first lectured on this, it took me 3 hours.
Oh, I see. Well,
and the problem is that it's so different from what people are taught in school that they can't swallow it.
Oh, well, these are physicists, so they
oh
were.
Well, in that case, they can't swallow it all.
Well, their biology is not always there.
Yeah. Okay. Convince them of anything.
Yeah.
Okay, well, I think we made a little progress in talking it around.
Yeah.
Okay. The question is what to do.
Right. Yeah.
Okay.
All right.
Okay, let's get on. Go on.
All right. Sounds good. Susan, do you have any updates for us? Are you working on anything?
No, I decided that the paper I was trying to duplicate ignored the instabilities in the tensegrities they were using and just mapped before it or graphed before the instability and then graphed afterwards and made a nice smooth curve and said, oh, there's instabilities. It's like, yeah, there are. That's So that's my update. They were cheating. Not exactly, but you know.
Right. They were selectively ignoring things that they didn't want to deal with. Yeah.
And then they said they were, but I would make the point by saying it was at this particular force or distance moved. It was at this point. Like, it's obvious. It's right there.
Right.
I get the graph every time.
Yeah. Morgan or Jesse, did you have any updates? Just wanted to give you some
yes,
space.
and I solved the problem, the 90-degree rotation problem, by placing the triangle in the negative y direction so that the y is actually pointing out of the plane.
Oh.
I was trying to explain that last time. I don't know if anybody cared, but anyway, I did solve that.
Good. So
now I'm going, well, these stress strain curves, you could make them point in any direction you wanted. That's not very good either, but because apparently if you make the top and the bottom and the bars infinite elasticity, then you get a J-curve, which is what you're looking for in a cell. So somehow cells have kind of an infinite elasticity, like over 200 gigapascals.
Wow.
I'm going, maybe if you include the myosin and the fact that they're actually contracting. I don't know. Anyways, it's a mystery. All I can do is just write up this.
Yeah.
Like, I want my graphs. I want to be done with it, and I want to write it up. I'm so over this project. It's just, let me do something else, please. Anyway.
Yeah. I know what you mean. Yeah.
Yeah. Oh well.
Yeah. Looks like Morgan and Jesse are okay. I didn't see anything. If you want to mention anything or ask any questions, please feel free to put it in the chat. So I'm going to go back to sharing my screen here. Or have I shared my screen? Oh, I did share my screen. Okay. Go back to sharing my screen. And I'm going to talk about this first. I get, and I know Morgan knows about this because he has the T-shirt. There's this organization, of course, called Fermat's Library, and they have a paper of the week. And every week they have like this. Fermat's Library is like a set of papers where people can build a library in this specialized PDF interface where you can mark up the document collectively. You can take notes on a paper or whatever. So that's where this is coming from. And this last week, they had a paper from John Maynard Smith in 1970. And he published a paper entitled Natural Selection and the Concept of a Protein Space. This proposes a simple analogy for the incremental process of adaptive evolution. His protein space analogy contains the basis for many central ideas in evolutionary genetics. So he likened the evolutionary trajectory of proteins to a simple word transformation game in this letter. This game, with the goal of evolving one word into another by means of a single letter substitution, serves as a striking model for protein evolution. Some of the work I've done with vehicles and composing organisms, there's sort of a quasi-developmental process. I illustrate this with these kind of word games, and I didn't remember that this was this sort of what this was, this exercise. But it's basically, taking like a string of letters, and it could be DNA or it could be like the English alphabet, which has 26 letters, and permutating those letters. But you start with like a word, and then you permutate the letters to make new words. But they have to be words that mean something. They can't be gibberish. They have to, have like a meaning to them. So that's the challenge. And so it's like in a protein sequence or even in a DNA sequence, where you have a string of letters, you're mutating them, but you don't want to mutate to something that's not functional. You want to mutate to something that's functional. So that's the idea. So you have 26 letters. You have 26 possible states in each position. If you have a string of 4, you have 4 of the 26 possibilities. But you don't, like I said, you don't want to use all those possibilities. You just want to use the possibilities that have some meaning. So usually what that means is that you can build a transformation path from one word to another by mutating one letter and then maybe mutating another letter. And it's a little bit artificial because you're kind of doing this consciously to sort of to hit words with known meaning. So an example would be like word to war. So you're mutating the last letter from D to E. Then the third letter is gore, or the third word is gore, which is mutating the first letter from W to G. And then the fourth word is gone, which is where you mutate the third letter from R to N. And then the fifth word is gene, which is mutating the second letter from O to E. So you go from word to gene in 5 steps. And you can map that out in a tree. Go ahead.
Bradley?
Yeah.
This is a little bit similar to a game we played as kids. Take a given word. How many words can you make from the letters in it?
Oh, yeah. Yeah.
In that case, you were restricted.
Yeah.
To the letters in the original word.
Oh, right. Yeah. Yeah. I mean, it has probably a lot of similarities to things that, and it's kind of a fun thing to do because you see kind of the similarities between things, and so yeah, it has some, there are other games that you can play with this, but so you can build a sequence, and then you can build alternate sequences. So it's not just one sequence. It's like you have multiple paths from word to gene, and you can just mutate different letters and get there in different ways. And so this is similar to the concept of a neutral space. And if you're not familiar with the neutral space, it's basically the space of possible genotypes that if you were to just let your system sort of go at random and explore different states, you would end up in like there isn't any sort of specific spot for mutation or any specific mutation rate. It's just exploring that space at a neutral rate, in a neutral way. Where would you end up? And so a lot of people characterize this with a hypercube, and they have different states in the nodes of the hypercube, and you have to traverse a path through the hypercube to get to the other side, and you have to count how many steps it takes you to get there. And so this is sort of similar to that as well. And you can look at optimality criteria and all that. And then there's this sort of analogy between functional proteins and words in English or words in any language. You have a finite alphabet. You can look at their the transitions between different words and build a space from that. And so that's sort of treating protein sequences and gene sequences as like a linguistic sort of dictionary. And so this is kind of this was from 1970, and then this is the paper here, where this is where the notes are taken here. So that people just pop notes in here, and I don't think this is the paper. I think I just took a screenshot of it, but this is sort of where he starts talking about natural selection and the concept of a protein space. So instead of thinking about it as a neutral space, he talks about natural selection, which would be where there's a directional imperative for selection to go to like the other side of the hypercube in that neutral space in a minimal number of steps. So instead of just exploring every mutational pathway, there's an imperative for natural selection. And if you think about like the sort of the artificial nature of this, where you go from word to gene, you're really imposing sort of artificial selection on this to say, I want to go to a certain location. Can we get there in a certain number of steps? And so that's the thing. Yeah, it is similar to a lot of games that people have played. It's very game-like, you know. You have a lot of states you can visit, and it's nice. But that's something in it that relates to what we were talking about before, because you're looking for things that are permutating, maybe for different cell types. You might have differentiation genes, for example, that all are sort of highly conserved. So you have this one process that unfolds in every cell type, and it just tells it to go one way, tells it to say differentiate now. Or you could have something that mutates a lot, and it has a specific code to say it's this cell type, or which is maybe less likely because it's through different, like throughout evolution, you'd have to really kind of target, or it'd have to be under high selection for certain cell types. So a little bit of a word on what hyperspaces are. So a lot of times, if you want to envision a sort of an evolutionary space that is subject to neutral processes or neutral theory, people have come up with this idea of a hyperspace. So I'm going to draw a simple example here where we have a cube, and this cube has a number of binary states. We have 8 nodes, and we characterize it with a 3-bit register, so we have this path from 0, 0, 0 to 1, 1, 1. So we're going from the bottom left-hand corner to the top right-hand corner of this cube. And the idea is that you can navigate across from node to node, and each node represents some mutation on 0, 0, 0. So you could have 0, 0, 1, 0, 1, 0, 1, 0, 0. Basically, it'll allow you to move to 1, 1, 1. So you can have each step as a mutational step, and it changes what they call this the phenotype. So a lot of these models have been developed in RNA, looking at RNA molecules and looking at their phenotypes. So a mutation in an RNA molecule is a change in the phenotype. It can change the conformational structure. It can change the message that's inherent in the molecule. So people also use hypercubes, and I think I mentioned this in the lecture, where you have a hypercube. So you have a cube within a cube, basically. Sometimes people call these tesseracts. And I think they used the word tesseract incorrectly in the Marvel series of comics, but whatever. So this is our hypercube, and it's basically a cube within a cube. So it has a lot more states, and you can explore more space with this. And there are other sort of mind-bending topologies that you can use. It gives you a larger number of states, but it also gives you this larger space to traverse. And the idea, though, again, is to go across the space, traversing it one step at a time, and then figuring out how many steps you need to take to get to your endpoint. Now, the other thing I want to mention is that, I talk about this as if like it's a magical process, but it's really about, what are the constraints that you impose on these models. So, when we talk about neutral processes, we're just basically talking about a system that explores that space at random. So this space has 8 possibilities, and so you can explore it at random. So you could start with 0, 0, 0, and you could move up to 0, 1, 0. You could go to this back left node here, and then down to 0, 0, 1, over to this bottom right node, and then over to 1, 0, 0, up to this front right node, and then to 0, 0, or to 1, 1, 1. And so that is 7 steps, which is not optimal, because what would be optimal would be to go from 0, 0, 0 to 1, 1, 1 in 3 steps. And you can go take 3 steps to get there. That's the shortest route. But in a neutral process, you might end up achieving that in 7 steps. It's just a matter of random mutation and not favoring any one site over another, not favoring any one strategy over another. And so and then natural selection is more reflective of this shorter path, or this optimized path from one phenotype or phenotypic state to another. The other thing I wanted to talk about is this. Go ahead.
Bradley, before you do, just brought up something interesting. Maybe one approach would be to say, take a particular cell type and get it for an organism that's been sequenced, and then pick another organism but the same cell type and see what's similar between them.
Yeah.
Okay, so you're looking at particular cell types across genera.
Right. Yeah, that would definitely yield an answer of some type. Or at least you'd have an idea of what to look for.
And has it been done?
Well, people have compared different species and cell types. I'm not sure they've asked that for that specific question, so they're probably not looking for, they might be looking for a specific functional gene, like maybe a muscle gene muscle specific gene in 2 species, but then you'd have to decide whether you want something that's closely related or not, because a lot of people look in model organisms, and it's like a mouse and a C. elegans, which isn't.
Yeah, well, in regard to this, there's some old work back from the 30s, well before the genes were discovered.
Yeah.
Okay, where you could actually take, let's say, fish liver and put it in a chick, or vice versa.
Oh, right.
And the liver cells would be incorporated into the liver of the organism.
I was thinking eye retina in eyes in vertebrates might be interesting.
Yeah. But that's a number of cell types, so it's different. It's hard to get a pure cell type out of it.
Yeah.
Cones and rods.
Well, yeah. I guess if you take a sample of tissue from any organ, you're going to get you'll get cell types of different levels of differentiation sometimes, and sometimes you'll get a single cell type, like in liver, versus if you were to sample in retina, you have a bunch of cell types jumbled up in there,
you do.
But it's so the data set you would get would probably not be one cell type. We'd have to work with the data we have available. You could do the experiment. We could maybe separate out different cell types, but maybe that's a good Those are of course classic experiments where you just take a cell type from one organism, you put them in another organism, or So.
Brad, maybe take your notes, organize them a little bit, and send them to me.
Okay, yeah, well. Yeah, I'm taking notes on this. Yeah.
There are a lot of ideas here, so that might be an approach.
So I think Susan threw a bomb earlier where she talked about physicists not having a lot of biology background, which is somewhat true, but there's this argument that biology is more theoretical than physics. And this is a newish paper. Yeah.
The biophysicists I talk to are definitely learning the biology for what they're doing, but yeah, you go off what they know, and these students seem to have some difficulty with that. So, well,
Yeah, it's a challenge to kind of cross,
cross
yeah,
yeah,
cross
and I
disciplines.
know because I've done this a few times. It's like,
Of course, history, for example, of molecular biology, it's physicists who led the way.
Yeah. So this is a paper. This is from 2013, actually. So it's about 10 years old now. And it's called "Biology is More Theoretical Than Physics," and so it's kind of an argument that we think of physics as maybe being really theoretical and biology not being so theoretical. But this paper makes the argument that the opposite is maybe true. And so the abstract reads, the word theory is used in at least two senses. One is to denote a body of widely accepted laws or principles, as in Darwinian theory or quantum theory, and to suggest a speculative hypothesis, which is two, often relying on mathematical analysis that has not been experimentally confirmed. So it's basically this widely accepted laws or principles that you have developed, and then speculative hypotheses. So this is just the way that people use theory. And then this is only two definitions. There are probably more. It is often said that there is no place for the second kind of theory in biology, and that biology is not theoretical but based on interpretation of data. So this is where often people will go and get lots of data but have no framework, or they may have a framework for it in maybe like accepted laws or principles, but they don't really have a lot of room for building speculative hypotheses that go just kind of beyond data mining and that sort of thing. So that's something that maybe it's sort of the way that the field works. I don't know, but anyways, here ideas from a previous essay are expanded upon to suggest, to the contrary, that the second type of theory has always played a critical role, and that biology, therefore, is a good deal more theoretical than physics. And so this kind of goes through some of these things that in a previous essay I pointed out the curious case of the enzyme substrate complex, which was widely used to understand enzymes before any enzyme substrate complex was shown to exist. So this is where you have this idea of this structure or this thing we were talking about with differentiation genes. You don't know if it exists or not. No one's shown it exists, but you kind of build a model and you say, this exists and I'm going to show that it exists. And in a way, that's how you kind of do biology, although, a lot of times people will, like I said, they'll collect data on, some, comparing two different species or, looking for all functional genes of a certain category or something like that. But this is where you actually build a sophisticated model of something, and then you kind of realize it with data. So Britton Chance, who brought these hypothetical entities into existence, was in no doubt that he was providing the first experimental confirmation of a theory. So this is that one. I think Britton Chance was also the person who developed FNIR, if I'm not mistaken. But in the intervening 30 years, biochemists happily used a theoretical entity because it was so useful and explained so much. Expediency overcame the kind of philosophical scruples that would make a physicist swoon. And so this is something where perhaps this is a marginal episode among enzymologists which can be glossed over in favor of the party line that biology is not, of course, theoretical. I claim that this is far from the case. In fact, similar episodes have occurred throughout biology involving some of its most important entities. And so he talks about the receptor and some of the equations that they developed to describe a receptor. And these are all theoretical things where you have like these constants and variables that you're kind of coming up with. This is what it should look like. So this is the Hill function. And so this is, just kind of something that had to be experimentally verified, but you build this model and then you eventually realize it. And so, it took 30 years for the enzyme substrate complex to become a chemical reality. The receptor took a good deal longer. So this is all, kind of thinking about theory and how you realize it in data. Related to that is this paper by Wallace Arthur, and this is from biological theory. So this is actually this is on Darcy Thompson's morphological transformations, issues of causality and dimensionality. This is, of course, we've talked about Darcy Thompson before, who did a lot of things with mathematical transformations and looking at organismal phenotypes and how they change across species. So if you take like a fish and you put it on a grid and you map the intersections of the grid to the landmarks of the fish, on the phenotypic landmarks of the fish. If you take a different fish and you put it on that grid, you warp the grid to match the landmarks, and you end up with this transformation between a straight line grid and a warped grid. And so this is interesting because Darcy Thompson was building these theoretical models, and it's something that is a nice tool, but also kind of is a theory of transformational change. So Darcy Thompson's drawing showing coordinated differences between the shape of an individual of one species and the shape of an individual of another have been reproduced and discussed countless times. However, while they can be widely regarded as inspirational, their interpretation in causal terms has been proven difficult. And there is as yet no consensus on the matter. So this is, these models are somewhat predictive, but they're also mostly inspirational. So it's not that we understand the cause of those transformations. We just understand that those transformations exist. And so we're not really at the stage of a predictive theory. We're just kind of at the stage of like this nice tool that can demonstrate these transformations, but we don't really know why they exist in that form. Here I approached these Thompsonian transformations from a particular angle, namely their dimensional insufficiency. I argue that this problem must be solved before the issue of causality can be considered. So a lot of times in theory we want to know about causality. We want to know if one thing causes another. And so, but maybe that's the wrong question to approach right away. Maybe you need to understand, what he calls dimensional insufficiency. So the Thompsonian approach, or this approach of looking at dimensional insufficiency, leads to the conclusion that Thompsonian transformations or morphological transformations, remember those transformations of the grid, have not taken place in evolution, and logically can never do so, because they involve the direct conversion of the adult form of one species into that of another. So this is really interesting. The whole idea of the Thompsonian grids is to show this transformation between, like if you want to represent an adult phenotype of one species versus the adult phenotype of another species, you look at these mathematical transformations and you say, well, you assume it must have gotten there in development. But we're not actually measuring that. We're measuring adult form versus adult form. So what he's saying here is that these transformations are all adult-adult comparisons. And so you can't necessarily say that these conversions represent what's happening, say, in development. So that's where you have this problem. You use a nice approach like that, but you also have to have this developmental aspect. So what he does here is he talks about developmental transformations, which are also important, and they would actually involve the phenotype developing along a certain trajectory. And that's really the whole business of EvoDevo, is to explain, like, if you have adult forms that are variable, and especially a trait that's variable, how that gets there in development, or what the developmental mechanisms are underlying that. So this is, in contrast, developmental transformations do occur in the short term. And so those developmental transformations don't necessarily show up in those grid transformations. Those grid transformations are sort of the end product of the developmental transformations. So you have to think about it in a different way. It's not that those transformations are going to explain anything on their own. They just show what happens when some X factor, which is developmental transformations, unfold. And so there are these developmental transformations that occur in the short term within the lifetime of a single individual. And evolutionary transformations occur in the long term in a context that can be described following Scott Gilbert as five-dimensional. Scott Gilbert's a developmental biologist who described these as five-dimensional transformations. I argue that these two kinds of transformation, developmental and evolutionary, have different causal agencies. I consider the possible nature of these agencies and, related to that, in the way in which Thompson's work connects with Darwinian evolutionary theory. So he really kind of lays this out in terms of a lot of what Darcy Thompson did in terms of the developmental biology, and that involves the expression of Hox genes and other things that, so this is an example of these graphs or these grids and putting different adult phenotypes on them, and you use different landmarks like the tip of these antennae or something to sort of as a landmark, and then you warp the grid accordingly. So if there's a larger distance between this left appendage and the antenna, that grid will warp accordingly. So it's shorter here and longer in the species, and then it diverges outward in the species. So there are a lot of ways you can transform these grids. But, this is something that is a nice tool, but it doesn't really explain the development or even the evolution, because the evolution would have to be of the development. And so this is really kind of getting at a lot of theory building and causation in some of these issues. And then, I'm not going to get into this too much today, but there is this this document, 101 Unsolved Puzzles in EvoDevo. And when I mentioned EvoDevo, or the evolution of development, this is like all these unsolved problems. So these are problems that we've kind of observed in biological systems, and we don't really have a theory of how they happened. So, there are a lot of puzzles involving origins, evolvability, the genome, symmetry, asymmetry, digits, behavior, nervous system. There's just a whole host of open questions here, puzzles, and they range from why did hair evolve in mammals, or heterodonty evolve in mammals, to things like, with symmetry, what's the cellular basis for monozygotic twinning, how does twinning occur in armadillos. So you go from the level of different traits to the level of things in specific species, to the cellular basis for epithelial fusion. So you have a process where you want to know the mechanism, what causes the loss of tissue at symmetric fusion planes. So you have specific sort of processes, scenarios in different processes. And so these are kind of interesting because they kind of go through a lot of unsolved puzzles that this author has posed. So this is these are numbers in brackets to note references as enumerated in quirks of human anatomy. For context and perspective on these topics, see quirks. Definitions of EvoDevo terms. So there's this book, Keywords and Concepts in Evolutionary Developmental Biology, which is a good book if you're interested to know kind of the jargon of that area of EvoDevo. They kind of have a lot of things on evolution and development. It's a good book for kind of the introduction to that area. And so they're kind of putting these together from these two sources largely. And I think it's a nice list because it gives all these different examples of different biological questions and potential theoretical questions. And it gives gives a nice diversity of things and how we think about theory and how we think about asking questions in biology.
Bradley?
Yeah.
I have two comments.
Yeah.
One is basically we have a bunch of software available, probably online now, for warping. And my question is, to what extent does warping do what Darcy Thompson transformations do?
I think there's a similarity in the math. So like a lot of times they're using the same sort of techniques. Where Darcy Thompson was using, doing it by hand or by like, but they.
Right. Do you get the same result? I guess is the question.
Yeah. Well, they don't have it as a grid. Usually, it's like if I take an image, I can warp parts of the image. So I have like these features in the image that I can pull apart or dilate or make smaller. And
you have parts that you think coincide. Use those for warping.
Yeah.
Right. Okay. The other comment is that this ignores Haeckel's old what do we call it?
Biogenetic law.
Biogenetic law, right? The biogenetic law. You see, if what Haeckel noted is that embryos at early stages all look alike.
Right.
Okay. I was joking with my wife's been watching some movies on people rescuing birds as eggs, and they can't tell what bird it is until it grows up.
Right.
Okay, because all early birds look pretty much the same. So joking, we should have a guide to baby birds with just one picture. They all look the same.
Yeah.
Okay.
Yeah.
So the idea that the transformations are reversible may be wrong.
Yeah. In the Wallace Arthur paper, I think he mentions Haeckel's work, and I don't know what I haven't read through what he says about it specifically, but
I mean the transformation from the adult back to the embryo.
Oh, right. Yeah.
May not tell you anything about the transformation of the embryo to another kind of embryo back to the adult.
Yeah. Yeah, that's true. And it's,
in other words, these transformations, how should I say, they're not reversible.
Right.
Okay. Okay, which screws up the whole thing in a sense.
Yeah.
I mean, if I gave you the transformation between very different adults, which have similar embryos, how on earth would you take them backwards?
Right. Like finding the common point, like just like reversing that warped grid and just finding the midpoint, or that wouldn't really tell you anything.
That doesn't mean anything. I'm not sure what the transformations between adults means a lot more than the transformation between embryos.
Right. Well, I think the point, though, too, is that the transformations between adults tell you something about the adult phenotypes, but that process from the embryo, or that process from an evolving, so, the common ancestry between them, or rather, what's going on in their development is a separate thing. So it's not, it may be responsible ultimately for that transformation, but we can't say that, we can say, okay, there's something that moved this here, moved that there, and it's a much more complicated process than that.
Yes.
Yeah. All right. Any other comments or questions? All right. Yep.
I'm going, no, don't have comments or questions. Just,
all right.
Thanks for the session.
No problem. Yeah.
Bye.
All right.
Okay, bye.
Talk to you later. Bye.
Bye.
Bye. Now I'd like to talk about two papers that one was the one Himanshu talked about, and the other one is something that I found that's related to this. So let's get into it. The first one is link prediction with persistent homology. This is the one that Himanshu talked about in the meeting. This is kind of moving from that microscopy and segmenting things and finding features to building nodes and then links. And actually, at that point, you need to predict your links, and you can use persistent homology to predict links. So this is what this paper is about. So this is, again, you have this underlying graph structure of the data, and you're trying to predict the links. So link prediction is an important learning task for graph structured data. So this is an interesting approach. What they're saying is that the data that you have is inherently structured as a graph. So it has connections, has interactivity inherent in the data. And so you need to find the links. You need to find the important links in this process, in the data set. So it's an important learning task. So we talk about graph neural networks and topological data analysis and graph neural networks and that sort of intersection. We need to think of link prediction as a learning task. In this paper, we propose a novel topological approach to characterize interactions between two nodes. So they're actually using a topological approach instead of maybe like a statistical approach, where like we saw in the hypercube example or the neutral space example, just simply associations based on the state of the nodes. What's the path from one node to another? Is it mutational? Is it something else? But this is a case where we don't necessarily have that natural guide. We have to predict the links from where the data, the association of the data, the association of the features with one another. Our topological feature, based on the extended persistent homology, encodes rich structural information regarding the multi-hop paths connecting nodes. So this is where, again, we have this cube, we have 0, 0, 0, we have 1, 1, 1. We have a path between them. We need to predict these, what they call multi-hop paths. This is a communication networks term, but basically having multiple sort of steps to getting from one point to another in the network. So we need to be able to predict these paths, and in doing so, we predict links. Based on this feature, we propose a graph neural network method that outperforms state-of-the-art on different benchmarks. As another contribution, we propose a novel algorithm to more efficiently compute the extended persistence diagrams or graphs. This algorithm can be generally applied to accelerate many other topological methods for graph learning tasks. So they kind of go through this. They show two example graphs here. So you have these multi-hop topologies where you go from one red node to another red node. And you can see as they get more complicated. So this is kind of analogous to our cube versus our hypercube example when I was talking about neutral spaces and that sort of thing. So this graph on the left, you have this multi-hop architecture where you want to go from one red node to the other. What are the intervening nodes? And then obviously what are the intervening links? The second one is a little harder because there's more interactivity. So in the first one, you have three alternate paths. In the second one, you have three alternate paths, but those paths interact in different ways. So that's what we have here. So there's a lot of data that we can capture and characterize here. So this is an example here in figure 2 of extended persistent homology. And so A is where they plot the input graph of the given filter function. So they're plotting this over time. So you have at different time points, these nodes appear and they are linked together in this way. The ascent in B, which is this, the ascending and descending filtrations of the input graph. So they have filtrations of it by time. So like at each time you get a new node that pops up. And so as those pop up, you get new links. So like, for example, you have node 1 appears at time 1, node 1 or node 2 appears at time 2, but you can't necessarily find a connection between node 1 and 2. At time 3, node 3 pops up, and of course there is a connection between 3 and 2, 3 and 1, but not 1 and 2. So it's not just connecting anything, it's connecting things as they appear, and then the evidence appears that they're connected. At T4, you have 1, 2, 3, and 4, and they're connected. And then at time 4, you also have the disappearance of 1, 2, and 3. So they disappear. You have 4 here. So these nodes, these blue nodes die. You get a red node 4. Then 4 becomes a blue node. You get a red node 3 that appears at time 3. And then time 2. So you're going. You're going up to time 4, and then you're doing a reversible analysis here. So this is where you're actually getting. Let's see. So the bars in the brown and blue colors correspond to the lifespans of connected components and loops respectively. So there's a lifespan, so things die off, things come back to life as you try to do this reversible process. You get this these connected components are all the components that are connected in a graph. So sometimes you'll have nodes that are sort of out there without any connections, but the connected component are the parts that are all sort of, you can go from any one node to any other node through the connections that have been built. The first 4 figures are the ascending filtration. The last 4 figures denote the descending filtration. So there's this filtration, like I said, you do a reversible filtration, you do a forward filtration. In the ascending filtration, FUV max, FUV, well, in the descending filtration, FUV min, FUFV. So they're basically using a different criterion for each one. They're using the maximum value and the minimum value. So you're getting 2 different ways of building this this graph, or building the links for the graph. You're using 2 different criterion. In the resulting extended persistence diagram, red and blue markers correspond to 0-dimensional and 1-dimensional topological structures. So these are, of course, the 0-dimensional points and the 1-dimensional points. This is death time and time. This is birth time and time. So you have nodes that are being born, nodes that are dying off, and you have this intersection. So high death times and lower birth times are 0-dimensional points. Higher birth times and lower death times are 1-dimensional points. So this is kind of their approach. Their contribution is, or actually in this paper we propose a pairwise topological feature to capture the richness of the interaction between a specific pair of target nodes. We compute topological information within the vicinity of target nodes, which is defined as the intersection of the k-hop neighborhoods of the nodes. It has been shown that such locally enclosed graphs carry sufficient information for link prediction. So they're able to boil this down to these local graphs. This carries enough information for link prediction. So you're basically evaluating small parts of the graph. It has enough information to tell you about the links, and then you can you find the intersection of these larger. They're sort of neighborhoods of multi-hop relationships, and you're able to predict the links from that, and then you're able to build up from those neighborhoods to a larger graph structure. We propose topological loop counting graph neural networks, or TLC-GNN, by injecting the pairwise topological feature into latent representations of the graph neural network. And so this achieves good performance computationally, and they use extended persistent homology. And this is the citation Cohen-Steiner, 2009, and that is here. So this is the paper, Cohen-Steiner et al., extending persistence using Poincaré and Leshev's duality. So this is the paper that they're drawing from. And so, in summary, our contribution is threefold. We introduce a pairwise topological feature based on persistent homology to measure the complexity of interaction between nodes. We compute the topological features specific to the target nodes using a carefully designed filter function and domain of computation. So they define their sort of topological features in their nodes very carefully, and they can predict links from that. So they have these neighborhoods, they have these this sort of birth and death process, and that's what they're using to sort of make these predictions. We use the pairwise topological feature to enhance the latent representation of a graph neural network and achieve state-of-the-art link prediction results on various benchmarks. So they do this in a GNN context where they run it as an algorithm, graph neural network algorithm. We propose a general purpose fast algorithm to compute extended persistent homology on graphs. The time complexity is improved from cubic to quadratic to the input size. It applies to many other persistent homology-based learning methods for graphs. So they're able to do comparatively well. And so I'm not going to go through the rest of the paper. It looks like they do a lot of highly technical work here, and we can talk about this later. Himanshu might want to present on some of this later, but this is a nice paper for getting, we're making predictions of links. Link predictions generally have there's some sort of strength of the connection, and then you're using a different criterion like a maximum value or a minimum value to sort of predict that link, whether it should be there or not. So this is a nice paper going over how to construct these, and especially in a developmental context where we have cells that are being born, cells that are dying, cells that are diverging from their ancestral cells. The descendants are diverging, and we actually have we actually have two connectivity networks of interest. We have a lineage tree which predicts which cells emerge from which other cells, so they're going to have an affinity, and a spatial network which predicts where they are in space, where they are in the embryo, and so forth. And so this is useful. I think this will be a useful sort of guide to some of these things. Another paper I want to talk about is transformers meet directed graphs. And so we talked about directed graphs, lineage trees as directed graphs, differentiation trees as directed graphs. And so directed graphs, again, you saw in the example with the differentiation tree. We have a mother cell, we have daughter cells, and we have this replicated, this sort of binary division. Sometimes you have things that are, have multiple cells originate from sort of an ancestral cell. But most of the time, and sometimes you have asymmetric divisions where you only have one daughter cell. But most of the time, this is all can be characterized as a directed graph. So we can use directed graphs, but we can also use transformers, which is a machine learning technique. And these things, as opposed to like maybe using graph neural networks, we can also use transformers and understand these, or use them in a similar way. So the abstract reads, transformers are originally proposed as a sequence-to-sequence model for text, but have become vital for a wide range of modalities, including images, audio, video, and undirected graphs. So these are, different types of input data. We have video, you might want to analyze audio or still images, but also undirected graphs, which are just graphs with interactions. Like we saw in the last paper, you just have these interactions you're trying to predict. Sometimes you're trying to predict maybe causal linkages, or sometimes you're trying to predict like ancestry. And so you're trying to do these things where you have some information about the direction of your graph. And so, however, transformers for directed graphs are surprisingly underexplored, despite their applicability to ubiquitous domains, including source code and logic circuits, and development. So we'll put that in there. In this work, we propose two-direction and structure-aware positional encodings for directed graphs. So they're doing two-direction and structure-aware positional encodings. So the positional encoding is like, it's like what's the position in the order. So is it, before or after, basically. The first is the eigenvectors of the magnetic Laplacian, a directional aware generalization of the combinatorial Laplacian, and 2, directional random walk encodings. So directional random walk is where you take a random walk, you take you take a step, you draw from a random distribution, you take another step, and so on and so forth. A directional random walk is that you have a certain direction. So you might randomize the direction, but you have to go in a certain, like forward, instead of, a true random walk, you could go any direction you wanted. And so you end up with these little clusters of movement around a central point. A directional random walk is maybe like all the way maybe to the top of the screen if you're doing a simulation, or to one side of the embryo or another if you're in a biological system. Basically, you're randomizing your movement, but always in one direction. And this is good for like directed graphs because that's what directed graphs are. They're just this, they proceed in a certain direction. Empirically, we show that the extra directionality information is useful in various downstream tasks, including correctness testing of sorting networks and source code understanding. Together with a data flow-centric graph construction, our model outperforms the prior state-of-the-art on the Open Graph Benchmark Code 2 relatively by 14.7%. So this is, so they talk about transformers. Transformers are used to generate solutions for competitive programming tasks, from textual descriptions for conversational question answering with the popular ChatGPT. So ChatGPT uses a transformer, or to find approximate solutions to combinatorial optimization problems like the traveling salesman problem. These are all standard benchmarks. Transformers have also had success in graph learning tasks, from predicting the properties of molecules to other things. While virtually all prior work focuses on undirected graphs, they talk about directed graphs here. And so the attention mechanism needs to become aware of the graph structure. So transformers use this attention mechanism. For example, prior work modified the attention mechanism to incorporate structural information, and I think that's the same paper. That's a different paper. Or to proposed hybrid architectures that also contain graph neural networks. And so this is where you have, the attention mechanism in the transformer works instead of on visual features. They're working on some of these aspects of graph structure. So this is, the way we're going to go forward in this paper. Another complementary option are positional encodings that are used by many, if not most, structural aware transformers. So these are positional encodings, which is where one thing comes first, then another thing. So this MIN paper actually is the properties of molecules. So they're using this structure-aware transformer for predicting molecules. And then there's this Muller paper, which is actually attending to graph transforms. So this is where they're actually applying attention to graph transformers. This is another example in Archive. This is the MIN paper. So this is transformers for graphs, an overview for from architectural perspective. It's also in the Archive. So these are different ways you can do this by merging attentional mechanisms and graph transformers and graph neural networks. Okay, so now we have, there's a motivation for building directed graphs. This is an example of the first eigenvector of magnetic Laplacian. Node size encodes the real value and colors of the imaginary value. So they have these different ways that they've implemented this. They have the sequence, the undirected sequence. So you have the sequence which kind of goes in a certain direction. It's like going from 1 to 10. The undirected sequence, which goes in different directions from a source node. A binary tree, which has splits between a single node and two different nodes. And then a trumpet, which is this thing here where you have a directed sequence, but you have a shortcut that cuts across. And so these are these are examples of sorting networks, but they're also this first eigenvector of the magnetic Laplacian. The goal is then to predict whether the sequence is a correct sorting network. Based on the sequence of operations, we can construct a directed acyclic data flow graph that models the dependencies between the operations. So every node that comes before has a set of dependencies. So every node that comes, after the node that comes before has a set of sort of dependencies it requires the node that came before it. And if you have something in a sequence like that, in a temporal flow or a temporal sequence, you always need the mother cell. The daughter cell can't just pop up on its own. And, there are a lot of properties of these cells that we're interested in, like, different gene expression patterns or different properties that of differentiation that make this an attractive approach, at least in terms of the data flow graph abstraction. So, moreover, we showed that ignoring the edge direction maps both correct and incorrect sorting networks the same undirected graph, losing critical information. So they make a number of contributions here. They make the connection between sinusoidal positional encodings and the eigenvectors of the Laplacian. They propose spectral positional encodings. They extend random walk positional encodings to directed graphs. They assess the predictiveness of structural or positional encodings for a set of graph distances. They introduce the task of predicting the correctness of sorting networks, a canonical ambiguity-free application where directionality is essential. They model a sequence of program statements as a directed graph and rethink the graph construction from source code to boost predictive performance and robustness. And then they do the benchmarking. So they have a number of different things they introduce here. Basically, they're trying to find the positional encoding through these kind of signal processing techniques. Then they have this eigenvector Laplacian, which is this graph Fourier transformation. So they're taking the graph and they're using a Fourier transformation on it. Then they have directional spectral encodings. They define that here. They define this in terms of directedness. So we next illustrate how eigenvectors of the magnetic Laplacian encode direction. For the special case of the sequence, the eigenvectors are given by this function. This corresponds to the cosine transformation type 2 with the additional factor that encodes the node position v. So the eigenvectors of the magnetic Laplacian also encode the directionality in arbitrary or directed graph topologies, or graph topologies that are directed but also arbitrary, where each directed edge encourages a phase difference in the otherwise constant first eigenvector between these two. And so basically what you have is you have this phase difference. So each direction edge is sort of a phase difference in this eigenvector. So then they talk about directional random walks on graphs, which you ought to often use to find these paths. We showed in the last paper where you go from one node to another, you're basically crossing the graph and you're trying to find these pathways. So to overcome the issue of only walking in the forward direction, we can additionally consider the reverse direction. Additionally, we add self-loops to sync nodes. This avoids that A might be nilpotent and ensures that the landing probabilities sum up to 1. So we then define the positional encoding for node V, and so then that's how they build those kind of random walks that are directional. They can, sort of convert that to a directional encoding. So one important point about directed graphs versus sequences, so we showed the example of the sequence where you go around in a circle. Each thing has something that it's connected to in a certain order, but that's not really a directed graph. A directed graph is where you have a branching process that is dependent in time. And so that's a little bit different than a sequence, where it's kind of obvious what the path is. And so an important implication is that each topological sort of the directed graph is an equivalent program, or a different ordering of statements that yields the same result. In figure 7, we showed the number of topological sorts over the sequence lengths P for type of compact and deterministically constructed sorting network. For such networks in the sequence length of just 8, the number of equivalent sequentializations already exceeds 1 million. So this is a pretty large combinatorial space. Note that in the worst case, a directed graph has n n factorial topological sort. So that's a big number. Therefore, representing directed graphs of sequences can introduce a huge amount of arbitrary orderedness. In contrast to the sequential modeling, a graph-based representation can significantly reduce the size of the effective input space. So that's just a note on some of these different applications. Then there's an application of sorting networks. So this is something that goes back to Donald Knuth. It's a certain class of comparison-based algorithms that have the goal of sorting any input sequence of fixed size with a static sequence of comparators. So this is a sorting network where you have these different lines, you're comparing between them, and out of that you can build a directed graph. It's a little bit hard to see kind of how this applies to development, but I think it has a nice, maybe a set of applications in kind of thinking about transformers and thinking about topological data analysis and how to connect all this with development.